I hope it's better when we start the class. Our today's class is regarding the DNA structure and its replication. And there are two things that I'm starting my sessions. The first one is the Richard Dawkins said that DNA neither cares nor knows. DNA just is, and we dance to its music. That means the DNA is a combination of the genes, and it can be gated from our families through the genetical roots. And the other one, DNA provides the music and all our behavior as well as related to the proteins, because all of our body proteins do have a gene on the DNA strand. And our cells and the environment provide the orchestra. And Craig Winter says this. When we start from the structure of DNA, deoxyribonucleic acid is polymers of deoxyribonucleotides, and they are attached with each other by the phosphodiester linkages. And each the deoxyribonucleotide is composed of deoxyribonucleotides and inorganic phosphate group. And this inorganic phosphate group is acting as a backbone for the growing chain of the DNA strand. Each deoxyribonucleotide is composed of the nitrogen base, as we have seen yesterday, and also there are the ribose sugars, ribose deoxy sugars. The nitrogenous base might be the purines, or it might contain the pyrimidines. The DNA synthesis is always taking place from the 5' direction to the 3' direction. The 5' end means we do have deoxyribose sugar in the 5th carbon molecule. If it is associated with the phosphate group, you can call it's a 5' phosphate end. And the other end of the DNA strand is on the second carbon atom, the third carbon atom of the sugar. It might be ended with the OH group. And that one we call it the 3' end. So the synthesis always is taking place from 5' direction toward the 3' direction. However, the phosphodiester linkage runs in the 3' to 5' direction, or in the opposite direction. As I told you, each polynucleotide DNA strand has two ends. One of the free ends should be the 5' phosphate end, and the other free end should be the 3' end. And this 3' end carried unreacted hydroxyl group. Okay. And the 5' end carried unreacted phosphate group. Primary structure of the DNA and RNA can exist in the single strand form, and we can see the next diagram. When you see the anatomy of DNA, you can see here on the screen. There is a ribose sugar, and there is a nucleotide base that it contains. When we count the carbon atom of the sugar, this is the first carbon, the second carbon, you can see on the screen. The third carbon is it, the fourth carbon, and the fifth carbon. As usual, the fifth carbon is bonded with the phosphate group, and it carried. So that's why we call this is a 5' end. When you come to the other end that can have bond formation capacity of the phosphodiester linkage is the 3' end. The first carbon, the second carbon has OH end, and if it becomes free, you can call it that it has a 5' end or 3' end. When you come to the RNA, it has also the same kind of linkage structure, except replacement of uracil in RNA by the thiamine. So, as I told you, this is a phosphodiester linkage. By the way, ester linkage ስንል ሁለት ካርቦኖች መሃል ኦ ስትኖር ነው ኦክሲጅን ሊንኬጅ ሲኖር ester እንለዋለን። እዚህ ጋር ስታዩት there is phosphate as a middle, and there are two oxygen molecules. That's why we can call it phospho is there, and the ester linkage are there. So it's a backbone of the DNA strand, as well as it can act as also a backbone for the RNA strand. However, the difference is that the DNA has 2 strands, however, the RNA is a single strand molecule. So specific sequences can be in there, nucleotides, the adenine, thymine, guanine, or cytosine nucleotides might be in the DNA. However, uracil will be present in the RNA. So when you can see here also, the diagram showing a similar manner. There are 2 types of diagrammatic representations in A and in B. When we see the 5' end is always the phosphate end, the 3' end is always the OH group end, and this is structural representation of the 1 strand of the DNA, and this one is normal line diagrammatic representation of the strand of DNA. So you can use either of this in order to indicate the structure. ከዛ ውጭ ለምን ይጠቅማቸዋል ይባል ሚሞራይዜሽን ነው። ስዕሎቹና ዲያግራሞቹ በተለያየ መንገድ በሆኑ ቁጥር ለማስታወስና ለማወቅ እንዲጠቅማችሁ ነው። So DNA chain with the nucleotide sequence is shown written in the 5' to 3' direction ነው። ከዚህኛው ላይ ካለው ከምታዩት A ላይ ለምሳሌ 5' direction to 3' direction. Always the synthesis is taking place in this direction ነው። ኋላ ላይ እንደምናየው as the DNA has 2 strands, proofreading የሚባለው አለ. The proofreading is taking place from 3' to 5', while the synthesis is taking place from 5' to 3'. And when you come to the bonding, mostly it's an hydrogen bonding. If you know about hydrogen bonding, is taking place between one of hydrogen-rich compound and the other, the most electronegative element. It might be oxygen or nitrogen. So between the 2, the hydrogen bonding can be taking place. Based on the Chargaff's role, adenine is always paired with thymine, or if it is in the guanine is always paired with cytosine. Adenine and thymine nucleotides have the capacity to form 2 hydrogen bonds to stabilize the DNA structure, because those are acting as additional linkages while the double strand of the DNA is formed. And guanine and cytosine, they have triple hydrogen bonds, or the capacity to form that one. Due to that reason, we can call it that. The double DNA strand that contains much more of guanine cytosine nucleotides has a stronger melting temperature than the DNA strand having AT linkages. Okay. ምክንያቱም melting temperature ማለት by the way, the temperature at which the DNA undergoes separation ወይም ደግሞ annealing እንለዋለን። separated የሚሆንበት ማለት ነው። unwinded የሚሆንበት ቴምፕሬቸር ሁለት DNA ስትራንዶች separated የሚሆኑበት ነው። ይሄ melting temperature ያኛው DNA ስትራንድ ብዙ ጓኒና ሳይቶሲን ሊንኬጆች ካሉት ከፍተኛ ይሆናል። ብዙ አዲኒና ታያሚን ያሉት ደግሞ ከሆነ ከዛኛው የቀነሰ melting temperature ነው የሚኖረው። or characteristics of DNA based composition, as I told you before also, double strand DNA, the number of adenine residues is equal to the number of thymine residues. This is why, because of the Chargaff's rule, okay, the linkage is formed between in 2, and we have seen yesterday that they do have a balanced amount in a strand of DNA. It should be controlled during the synthesis of the nucleotide base. እዛ ሰዓት ላይ ኮንትሮል መደረግ አለበት። ሁለቱም እኩል መሆን አለባቸው ሲሰራ በሚያስፈልገው መጠን ማች ስለሚያደርጉ ማለት ነው። Further, number of guanine and cytosine residues should be equal for the synthesis. ኋላ ላይ ግን ብዙ ጊዜ ምንድነው as corollary sum of the purine residues is equal to the sum of pyrimidine residues መሆን አለበት። ይሄንን ያለን ሩል ትላንት እንዳየነው ማለት ቻርጋፍስ ሩል ነው የሚለው adenine plus guanine is equals to cytosine plus thiamine, the Chargaff's rule. Because complementary chain ዚህኛው ላይ እና አንደኛው ቼን ላይ እና አንደኛው ቼን ላይ ደብል ስትራንድ የሚፈጥሩት ላይ ያሉት ኒውክሊዮታይድ ሲኩዌንሶች ኮምፕሊመንተሪ መሆን ስላለባቸው የአዲና ጓኒን ቁጥሮች they should balance with the cytosine and thiamine. So the DNA from different tissues of some species have some base composition. So the base composition of DNA varies from one species to another one. Okay, genetical ስለሆነ ነው። ነገር ግን አንዳንድ ተመሳሳይ ስፒሺስ ላይ ስናይ the DNA base composition is similar. For example, if for the human beings we do have only 4 nucleotide base sequences, adenine, guanine, thymine, and cytosine, and those are similar match for all of the human beings ማለት ነው። የተለያየ ሲኩዌንስ አቀማመጣቸው ቢለያይም አደራደራቸው ቢለያይም ለሁማን ቢንግስ እነዚህ ነው ያለው። ሌላም ስፒሺሶች ቢሆን የዲኤንኤ ሲኩዌንሲንግ አቀማመጣቸው ጂኖች አብዛኛውን ተመሳሳይ። ለዛ ነው በአንድ ስፒሺስ ውስጥ ተመሳሳይ አይነት ስትራክቸር፣ ፊዚካል ኮምፖዚሽን ሁሉ ነገር የሚኖረው። DNA of widely different species have different base composition. አሁን እንዳልኳችሁ ነው። ምሳሌ the base composition of the human DNA is totally different from the base composition of another species. When we see that the fish, for example, it's totally different base composition ያለን እነዛ base compositionዎች different መሆናቸው physically የተለያዩ biochemically የተለያዩ changes እንዲኖር ያደርጋል። So when we see the Chargaff's rule, it was first identified in 1940, and it says that the DNA base compositions differ in species, and also different tissues of the same species have the same base composition. Different tissues ነው። ምሳሌ አንዳንዴ DNA ምርመራ ላይ ካያችሁ ቼክ ሲደረግ ጸጉር ሊወሰድ ይችላል። አንዳንዴ ካያችሁ ኤኒ ፓርት ላይ ፍሉዶች ላይ ሊወሰድ ይችላል፣ ከሴረም ሊወሰድ ይችላል፣ ስፐርም ላይ ሊወሰድ ይችላል፣ የተለያዩ ነገሮች። ግን by any means if the DNA is identified, it has similar composition in the organs. ብዙ ጊዜ አንዳንድ ፊልሞች ላይ ይሄ ልጅን ቼክአፕ ለማድረግ ምናምን ሲጠቀሙ አያችሁ በየትኛው ሚስ ከየትኛው ቦታ ዲኤንኤ ወስደው ያገኙት ከዛ አንደኛው ሳምፕል የሚሰጠው ሰው አለ ሁለቱን ኮምፔር ሲያደርጉት አይደንቲፋይ ይደረጋል because we will see on the synthesis of the DNA that one of the parental strand DNA is obtained from the mother, the other parental DNA strand is obtained from the father. Then the new DNA strands are synthesized by using those parental strands as a template. So DNA base composition does not change over time. I told you that one. Adenine is always equals to thymine, and guanine always equals to cytosine, and they can make a pairing. Those all are under the Chargaff's rule. When we see the differences between DNA and RNA, DNA, the sugar motif deoxyribose, we have seen yesterday that on the second carbon of the ribose sugar, if the OH group is present, the sugar what you call it, it's ribose. On the second carbon, if the OH group is reduced by the catalytic activity of ribonucleotide reductase enzyme, it becomes hydrogen, and you can call it, that's the deoxyribose sugar. When we see the DNA, uracil, it's a pyrimidine base, is usually absent, however, it's present in the RNA. DNA is double-stranded, while RNA is a single-stranded. The sum of purine base is equal to the sum of pyrimidine bases in the DNA. While in the RNA, the sum of purine bases is not equal to the sum of pyrimidine bases. So are also the differences. The other difference that we have seen is DNA is resistant to hydrolysis by alkali because absence of hydroxyl group on 2 carbon atom of deoxyribose. Yeah, so we can call it it's deoxy ribose sugar is there. Then in RNA, because of presence of hydroxyl group on the number 2 carbon atom of the ribose, it can easily be hydrolyzed by the alkaline condition. And the other one, so this is, by the way, the major concept. In the solutions, the RNA is easily susceptible to be undergoing hydrolyzing by hydrolase enzymes, while the DNA has much more resistance in such a manner. DNA bases are not modified, while the RNA base is modified, and as you know, uracil is present. It can be DNA doesn't have any catalytic activity. However, RNA has a catalytic activity. Yesterday we said that RNase is there. RNase is a small fragment of RNA that can have a catalytic activity. The foundation of RNA has also changed the description that says all enzymes are proteins. Nowadays we are saying all enzymes are proteins except RNase. Okay, because an RNase is a small fragment of RNA that has a catalytic activity. When you see the DNA, usually not subjected to degradation in the cell. However, RNA is subject to degradation in the cell by the nucleases, or they do have also hydrolase activity. Because, as I told you before, it might be hydrolyzed in the cells as the ribose sugar as free OH second group, free OH group on the second carbon atom of the sugar moiety. It makes it very susceptible for nuclease activity. When you see secondary structure of the DNA, it is performed when two strands of DNA are paired together. That means DNA naturally, it's a double-stranded molecule. The two strands are joining up together and they form the helix. So the chains are paired in anti-parallel manner. That means if one of the chain has 5' to 3' end, the other chain has 3' to 5' end. No, anti-parallel nature. If in the same direction, አንድ አይነት ቦታ ላይ ጫፋቸውን ስንመለከት አንደኛው ጫፍ ያለው 5' end ከሆነ የሌላኛው 3' end ነው የሚሆነው ማለት ነው. So the bases are sticked in the inside of the two strands by hydrogen bonds, and there are also other types of linkages which are making such a stabilization. The negatively charged phosphate group and the sugar units expose themselves to the outside of the chain and they are acting as a backbone, especially the phosphodiester linkage. The two strands of DNA coil around a single axis forming right-handed double helix. Right-handed ስንል እንደነው እጃችሁ ለምሳሌ እንዲህ ብታሽከረክሩት ወደ ቀኝ ጋር ብታሽከረክሩት ወይ ቾክ ይዛችሁ ወደ ቀኝ ያሽከረክራችሁ ሄሊክስ ብትስሉ መጨረሻችሁም ወደ ቀኝ ነው የሚሆነው. ሶ ዲኤንኤ የተጠቀለለበት ሄሊክስ ፎርም ስለሚያደርግ ወደ ቀኝ እንዲየዞረ ነው። አስቡት ወደ ቀኝ እየተሽከረከረ ማለት ነው. So most of the time it's right-handed, no? The overall structure resembles the twisted ladder. ወይም ከዛ ውጪ በቀላሉ ዲኤንኤ ስትራክቸር ለመስራት ሁለት ሽቦዎች ይዛችሁ እሱን እየተቀለላችሁ ስትሄዱ ማለት ክፍተት እያደረጋችሁ it can show the DNA strand helical formation ያሳየናል. As you know that the most common stable structure of the DNA is identified by the Watson and Crick, and we can call it the Watson and Crick model DNA structure. It is a right-handed, and as I told you, anti-parallel chains are forming this in double helix. The Watson and Crick form of the DNA is called that BDNA. BDNA. ምክንያቱም A form of DNA ሳርተ Z form of DNA ምንላቸው አሉ. The most stable form in the cell, in the cytoplasm as well as in the cellular condition is the BDNA. ለዛ ነው ብዙ ፖፑላር ነው. The BDNA has a shape structure rules አሉት. For instance, as you can see here on the structure, it has a major groove ምንለው አለው. Major groove ማለት twisting ሲሰራ ጥርሶቹን በላው አንዳንድ ጊርስ ላይ ጥርሶቹ ላይ ማሽን ላይ ያሉት እሱ እነዚህ ጥርሶቹ እዚህ ጋር የምትታየው as you can indicate on the screen the major group has a larger gap and the minor group has a minor gap ማለት ነው ከዛ ውጪ they do have also rules for the nucleotide sequence pairs that are present within one round of the helix. So sugar phosphate backbone and the hydrogen bondings are the two measures for stabilization factors. But however, there are additional stabilization factors of the DNA double helix structure. Misali hydrophobic interactions by the different molecules ነው ከዛ ላይ ኤሌክትሮስታቲክ ኢንተራክሽኖች ይኖራሉ all those have their own influence for the strong stability of the DNA structure as I told you before the right handed እጃችሁን ወደ ቀኝ ያሽከረከራችሁ ቀኝ እጃችሁን ስትሄዱ ወይ ቅድም እንዳልኳችሁ ሽቦችን ይዛችሁ ለሞዴል ሁለቱን አርጋችሁ ወደ ቀኝ እየተቀለላችኋቸው ብትሄዱ as you can be indicating on the or as it can be indicated on the screen right handed ነው ወደ ቀኝ ነው minor group ይችኛዋ ነች ትንሿ ጥርስ ነች major group ወይም ትልቁ ጥርስ ለምንለው ፎርምድ ይሆናል in a full helical ለምሳሌ full ማለት አንድ turn around 36 አንገስትሮን ነው ይላል ለእንግዙ ዘን ወደ ጎን ያለው ደግሞ ስፋት ይሄ ዲያሜትሩን ማለት ነው 20 አንገስትሮን ነው this is the form of DNA the most stable form in the solution ያልኳችሁ so the double helix 3.4 angstrom ነው ብለናል ቤዙን the hydrophilic phosphodioxyribose backbones face out ነው ወጭ በኩል ይወጣሉ ቅድም እዛ ጋር አይተናል especially the phosphodiester bond is acting as a back strong bond no and the bases found on the inside of the helix there are 10 residues per turn no one turn እዚህ ውስጥ ያሉትን ቁጥራቸው እስከ 1 2 3 4 5 6 7 8 9 10 ነው ካያችሁት ስክሪኑ ላይ በዚህ ኢንዲኬት ላደርገው 1 2 3 4 5 6 7 8 9 10 ነው የሎች ስክሪኑ ተከትላችሁ ካያችሁት so this is one turn ነው አንድ turn ነች so in one turn there are around 10 residues of nucleotides ማለት ነው and also in one turn we can get one major group and one minor group no this is the most stable form of bDNA Can you see the chemical properties of the nucleic acid? As you have seen, the backbone it is hydrophilic, and the hydrophobic bases are there. As we have seen, they come from the hydrogen bond. So the base stacking has also additional force, what we call it, the van der Waals and dipole-dipole interactions. Van der Waals and dipole-dipole interactions are helping for making much more stable and strong form of the bDNA structure. So the Watson and Crick, they have proposed a double helical model of DNA, and it has having the following important characteristics of features. የመጀመሪያው ምንድነው? As I told you before, the two helical polynucleotide chains are coiled around a common axis. ብዙን ጊዜ አንዳንድ ዩቲዩብ ቪዲዮዎች ላይ ገብታችሁ DNA replication ብላችሁ አሁን ስፔሻሊ 3D 3 dimensional animation ያዙ ቪዲዮዎች አሉ አንድ አክሲስ አለ እዛ አክሲስ ላይ ነው ፎርሜሽኑ የሚኖረው or DNA strand formation እነዛን ስታዩት more clear ያደርግላችኋል ብዙ አሉላችሁ so the two helical polynucleotide chains they are coiled on the 6th axis and the chains run in opposite direction on antiparallel ቅድም ብለናል the two antiparallel polynucleotide chains are not identical but they are complementary. አንድ አይነት ሊሆኑ አይችሉም። ቅድም እንዳልኳችሁ አንድ ደብል ስትራንድ አለን ይሄን ላይ እና ምንድነው አንደኛ ሲሰራ ፓረንታል ዲኤንኤ ስትራንድ ሲኖር እዚህ ጋር እዚህኛው ላይ ስትራንድ ያለው ኮምፕሊመንተሪ እንጂ አንድ አይነት ሊሆን አይችልም። adenine is paired with thymine, guanine is paired with cytosine, is based on the Chargaff's rule. Hence, not complementary nature. ግን ተመሳሳይ ከሆኑ ግን they cannot form a link based on the Chargaff's rule ማለት ነው. The purine pyrimidine bases are not the inside of the helix. The phosphate and the deoxyribose groups are on the outside. They are on, sorry. The purine and pyrimidine bases ቅድም እንዳልነው ሃይድሮጅን ቦንድ የሚፈጥሩት በውስጣዊ በኩል ሲኖሩ phosphate and deoxyribose groups ደግሞ they are facing towards the outside side in order to act as the backbones. Below, in the Watson and Crick model of the DNA, the diameter of the helix is 20 angstrom, and its adjacent bases are separated by 3.4 angstrom. We already said that one. Helical structure repeats after 10 residues on each chain. No. ቅድም አይተናል አይደል አንድ ዙር ውስጥ አንድ ተርን ላይ እንደዚህ ሞሉ ተርን ያልነው ላይ there are around 10 base residues አሉ ብለናል. The two chains are held together by the internal hydrogen bond for stabilization. ሌሎች ተጨማሪዎች van der Waals interaction and dipole-dipole interactions are also existing ብለናል. Adenine is always paired with the thymine, and guanine is always paired with the cytosine. It's a Chargaff's rule. And adenine and thymine are paired by 2 hydrogen bonds, and guanine and cytosine are paired by 3 hydrogen bonds. The double helix is stabilized by interaction between the stacked bases of the same strand, and also the Watson-Crick model of DNA is also referred B form of the DNA, because there are other types of DNAs, the A form, Z form, that we will see. Okay, so the B form of DNA is the most stable in one under physiological condition. In the solution case, I told you. However, the mitochondrial DNA has some difference with other forms because of its circular structure. And the mitochondrial DNA can be formation of zDNA and cDNA, which can be performed during either replication or transcription. When we see the property of the 3 forms of the DNA, we can see in the 3D form, the B form, the Watson and Crick structure, and its standard form, we can call it. Most stable under physiological conditions, we already mentioned the properties of this type. The A form of DNA, as you can see on the structure, it's a compressed one. So we can call it, it is a dehydrated form. Most of the time when the dehydration is taking place, it might cause molecular weight reduction as well as size reduction. You can see an example of injera and dried injera, dirkosh. The difference between the two is the weight of water. When water is removed, the dehydrated form becomes what you call it dirkosh. Size and weight difference such like that. The dehydrated form of the DNA is this is the A form, that's a compressed one. The Z form, it follows the left-handed circularization. ለምሳሌ ቾክ ይዘን ወደ ግራንድ ሄሊካል ስትራክቸር እየሳላችሁ ትሩ መቻችሁ ሌፍት ነው አይደል? እዚህ ዲያግራሙ ላይ ስታዩት ሁለቱ ራይት ሃንድ ናቸው የተጠቀለሉ ነው የሄዱት። ስታዩት ኤን ፎርም እና ቢ ፎርም። ዜድ ፎርም ግን ስታዩት the rotation it's towards the left side ያስታውቃል አይደል? እንደምታዩት so this Z form of DNA it is expected to have play a role in gene regulation. However, the molecular level studies are not completed now. They may not be completed for a lot of years. So it makes them very interesting. Always አዳዲስ ነገሮች እየተገኙበት ይሄዳል። ብዙ ያላለቁ ጥናቶች ስላሉ ዓለም ገና እዛ ደረጃ ላይ ስለሆነ ማለት ነው። When you see structural stabilization of the double strand of the DNA, electrostatic interactions are also there. The negatively charged phosphate group, they are located at the outer surface of the strand, where they can neutralize by the histones and cations. The cations, by the way, might be present in the solution, or they might be present as a cofactors in the magnesium, basic condition. Histones are the proteins which can be used to complex with the DNA. Histone proteins እንደምታውቁት carboxyl እንዳላቸው amino እንዳላቸው. Histone protein amino end, it can form a link electrostatic interaction with the phosphate group of the DNA. ከዛ እንደምንድነው ይሄ histonesን የማያቸው አንዳንድ ሜትር እቺ ቱቦ ሜትር አይታችኋት እስቲ እየተመዘዘች የምትወጣው. What is the advantage of the cover ብትባሉ? It's acting as a shelter and rounding material for the meter ማለት. Such like that histone proteins, they can protect the DNA and they can act as to keep it is binding with it. So the polyamines are also there. They can form an electrostatic interaction in order to stabilize it. When you see the base interactions, hydrogen bonds between the bases are there. I told you that. The dipole-dipole interactions as well as the van der Waals interactions. So the stacking interaction between the planes of adjacent base pairs, what you call it, the van der Waals forces, dipole-dipole forces. So when the question comes in the way that what holds DNA strands together, the two strands of or double strand DNA are held together by the number of weak interactions, as we mentioned. Hydrogen bond, stacking interactions are there, hydrophobic effects are there. So those are the major ones, the property that we have seen, especially in the B form of DNA is expressed in this diagram. Major groove, the minor groove, 3.40 nanometer of the major and minor groove. The diagrammatically expression የተለያየ ስለሆነ ባክቦኖቹም ተገልጸዋል. sugar phosphate backbone አለው. as a middle there is an hydrogen interaction between the base pairs. So as I told you before, what happens to double strand DNA when the temperature is raised? It will cause the local unwinding of the double strand of the DNA. And the temperature at which the DNA strand starts to unwind is called the melting temperature. So it occurs preferably in the regions where the two strands are held together less strongly. That means it is occurring in a faster rate where adenine and thymine rich nucleotide bases are much more in the strand. ምክንያቱም ሁለት hydrogen linkage ስላላቸው. So in the regions the strand is separate to form a bubbles of the single strand region. By the way, DNA replication process አሁን ስንሄድ double strand area የሚከፈትበትና bubble formed የሚሆንበት area is rich in AT region ይባላል. adenine thymine pairing region ብዙ የሆነበት region ነው. ይሄንን ደግሞ የሚመርጥ ኢንዛይሞች ፕሮቲኖች አሉ። ያንን ኤሪያ ሴሌክት አድርጎ እዛ ጋር በብል እንዲፈጠር የሚያደርግ. The DNA sequence in this region is enriched, as I told you, with adenine and thymine pairs. And in guanine and cytosine rich region, strands are held together by 3 hydrogen bonds, and they are more strongly than the AT rich regions. So when the temperature increased, the base unstack and hydrogen bonds break. It might cause the total separation of the two DNA strands. No, denaturation. However, if it is limited amount of melting temperature, it causes the separation of DNA strand around the etheric regions. The other one, the breathing of DNA, DNA strand can also undergo breathing by the nature ማለት ነው። ማለት ትንፈሳት አይሆን you can call it the breathing. hello
ሄሎ ሸፊና ይሰማል።
So even at normal cell temperature the DNA breathes. Breathing means it can undergo separation and rejoining up together by the temporary and reversible unwinding of the local regions. And the AT-rich regions are more likely to undergo a breathing than the CG regions. ይህም በቃ ዋነኛው ምክንያቱ ሁልጊዜ የሚሆን ኤቲ ሪጅኖች ሁለት ሃይድሮጅን ቦንድ ስላላቸው በሁለት ስትራንድ መካከል ሲጂ ሪጅን ደግሞ ሶስት ሃይድሮጅን ቦንድ ስላለው ስትሬንግዙን ስናይ የኤቲ ሪች ኤሪያስ ዊክ ነው የሚሆነው። This is one of the reasons why transcription initiation bubbles and DNA replication origins are much more of the AT region. እዛ አካባቢ ላይ ነው በብሎች ኤቲ ሪጅን ላይ ነው በብል ተፈጥሮ እዛ ጋር ዲኤንኤው ተከፍቶ እዛ ጋር አዲስ ሪፕሊኬሽን ለማካሄድ የሚቀለው። So it is easier for the proteins, that means RNA polymerase and origin binding proteins are there which are participating in the DNA replication process and to create the locally unwound regions. When we see the nucleic acids chemistry, the first one becomes DNA denaturation. DNA denaturation or melting. When the temperature is raised more than the normal rate, then we can call it that. Both the DNA strands will be separated totally and they becomes they undergo annealing እንለዋለን። separated ሆነ ማለት the DNA becomes non-functional in the single strand ከተቀመጠ። So duplex DNA molecules are denatured, that means they melted at extreme pH and at high temperature. So denaturation means it's melting, single strand DNA formed ሆነ ማለት ነው from double strand DNA. However, the DNA might undergo renaturation or annealing process. Annealing ሊያካሂድ ይችላል ተመልሶ double strand ሲሆን it becomes functional ማለት ነው። As I told you before, it has a melting temperature, the temperature at which half of the strands is denatured depends on the pH, ionic strength, or it might be due to associated with lower melting temperature, we don't know. The size in the base composition ማለት ነው። ምክንያቱም ቤስ ኮምፖዚሽኑን ማተር ያደርጋል የምንለው ሃይ ጂሲ ቦንድ ባለበት ቤዝ ኮንተንት ውስጥ ሃይ ቴምፕሬቸር ይፈልጋል ሜልቲንግ ቴምፕሬቸር። እንደገና ብዙ ኤቲ ሪጅን ያለበት ከሆነ the melting temperature becomes less ማለት ነው። So initiation site for DNA replication and transcription are often they are rich in adenine thymine base pair regions. We will see in the next. In the bases interacts through hydrogen bonding forming specific base pair. So the double helical DNA is here. It can undergo denaturation. It can be separated because the single strand DNA is not functional. And when the favorable conditions are present, separated DNA strands can undergo annealing or renaturation. This is a partially denatured structure of the DNA that's indicated. However, if the temperature is higher and the pH is also much more higher, such like that of the protein environment, DNA might also undergo denaturation. So the partially denatured DNA can undergo total separation of the strands, and we call this condition is totally denatured DNA. If it undergoes association, it can be returned back to the partially denatured DNA, and after it can undergo so the totally functional DNA structure of double strand. When you see the denaturation, the melting temperature. Misali, which one could you expect is showing the DNA strand that has much more of AT-rich regions? የትኛው ይመስላችኋል? The one which is indicated by the blue line or the red line? Hello? Are you with me?
Yes, teacher, ይሰማል።
See, which one could you expect? Which one of the line indicates the DNA strand having much more of AT-rich regions? Which one indicates the DNA strand having much more of CG regions? Okay, I'm looking for, okay, blue line. Students are replied on the messaging. Yes, the AT region, much more region is the blue line. It's unquestionable because the melting temperature is around average is around 84, like that. However, in the red line, it indicates the melting temperature is increased up to 87, like that region. So, or 85, the average. It shows that more CG regions require much more higher melting temperature. In the DNA strand, having less CG regions require less melting temperature. As you have seen yesterday also, the central dogma of molecular biology, it involves the three major reactions. From the parental DNA, there is a synthesis of new DNA, the replication process. From the synthesized DNA, the messenger RNA can be synthesized by the transcription process. This means the nucleotide sequences on the parental DNA will be converted into nucleotide sequences in the messenger RNA strand. Then the last process of the cellular signaling response in protein synthesis translation can be taking place from the messenger RNA. So when you see the flow of genetic information, the central dogma of molecular genetics, in our body, most of the animals, as we have seen before, normal processes, the replication, transcription, and translation. However, in some microorganisms, majorly in the viruses, for instance, we can see the HIV virus. RNA is a carrier of genetic material for those viruses. Entering into the host cell, they can undergo the reverse transcription process in order to synthesize the DNA strand from the parental RNA molecule. If once the DNA strand is synthesized, they can undergo the further transcription, translation in protein by using the host process of mechanism, and they will produce the viral proteins. So this is one of the mechanisms for the HIV virus. That's why the drugs like retinol and adenine. Hello. Hello.
ሄሎ ቲቸር፣ አሁን ትንሽ ጸጥ ብሎ ነበር። አሁን ተመልሷል፣ ግን ይሰማል። አሁን ይሰማል።
Okay. So that's why the drugs retinol and adenine, they do have reverse transcription inhibitory activity. Rather than that, they do have also a protease inhibitory activity that we will see on the next slide. So in some viruses, I told you, RNA functions as a genetic material. I told you also. And the reverse transcriptase was found in 1970. And after that, the reverse transcription process is included in the extended dogma of molecular biology. Right? What I told you before is, during the synthesis of DNA, There are two strands, by the way, they form the helix. One of the strands is acting as a parental strand. Based on that one, the new daughter strand can be synthesized. And from the other parental strand, the new another daughter strand is synthesized. ይሄ ነው ብዙ ጊዜ የሚሆነው አሁን ከቤተሰቦቻችን አሁን በመልክ ልንመሳሰል እንችላለን የተለያዩ ከእናት ከአባቶች አንደኛውን ስትራንድ ከአባታችን ፓረንታሉ ነው ይመጣል based on that one we can synthesize our body can synthesize our new DNA strand complementary ነው ማለት ነው እኛ ጋር አንዱን ስለምንወስድ አንደኛው ስትራንድ ደግሞ ከማተርናል ይወሰዳል based on that one we can synthesize a complementary chain new DNA structure. ስለዚህ complementary ስናይ the newborn DNA structure one of the strand has all nucleotide sequences of complementing with his maternal DNA ማለት ነው ምሳሌ እናትየው ላይ ያለው TCACGATC ያለ ሁሉም እንደዚህ የሚሄዱ ኒውክሊዮታይድ ሲኩዌንሶች ከሆኑ የዛ ኮምፕሊመንታሪ ሁሉ አንድ አይነት ሆኖ የሚገኘው ልጅ ዋጋ ብቻ ነው ኦኬ አባቱም ከሆነ እንደዛው ካልሆነ አንዱ እንኳን ሲኩዌንስ ሊቀየር ይችላል ማለት ነው በዚህ መልኩ it can be using as indicator for the family relationship especially ብዙ ጊዜ አባትነት ነው አይደል እናትነት that's why we can call it the DNA replication is semi-conservative half of the parental DNA molecule is conserved in each new double strand DNA or one of the strands of the paternal DNA might be available in the daughter molecule one of the maternal DNA strand sequences all are available in the daughter molecule ግማሹ በአንደኛው ሳይድ ግማሹ በአንደኛው ሳይድ ይሄዳል so double strands can be formed ማለት ነው when you see the significance the genetic information is ensured to be transferred from one generation to the next generation with high fidelity because of the semi-conservative replication process nature of the DNA structure when we see the features of DNA replication we already said that it is a semi-conservative process as it carries half of one of the strand from the parental and one of the newly synthesized strand based on that one and the other strand from the parental strand and as well as other newly synthesized strand based on that one so DNA replication is semi-discontinuous what does it mean we will see on the next now that DNA replication is taking place in the area where the replication fork is formed. Fork when shuka size desire. Synthesis ሲካሄድ አንደኛው ከተነጣጠሉ በኋላ አንደኛው DNA parental ተወስዶ አዲስ ይሰራል. By the way, the synthesis is taking place by the DNA polymerase 3 enzyme. And synthesis is taking place in two directions now. The major toward the center of the fork, or what you call it, it is the synthesis of leading strand. This synthesis is continuous. What the synthesis is Mikhail. The other direction, what you call it, the synthesis against the center of replication fork, no, or lagging strand. The lagging strand is synthesized in the fragment form. Those fragments are called Okazaki fragments and they have got a name after the first scientist to identify those fragments, Okazaki, the Japanese scientist. And DNA replication is bidirectional, as I told you. That's why leading synthesis in the other opposite direction synthesis, a lagging synthesis. So when we see the requirements for the duplication of both of the DNA strands, there are all deoxyribonucleotide triphosphates. That means deoxyadenosine triphosphates, deoxyguanosine triphosphates, deoxythymidine triphosphate, and the deoxycytidine triphosphate. እዚህ ጋር ዲያግራሙ ላይ ለምታዩት ኢንዲኬት ማለት ፎርክ ይመስላል አይደል እንደዚህ እዚህ ጋር ሴንተር አለች can indicate on the screen can be indicated on the screen ከፎርኩ towards ወደ fork direction synthesis አለ ቅድም እንዳልኳችሁ the synthesis is taking place from 5 prime to 3 prime direction against ደግሞ ይካሄዳል በዚህኛው ሴሌክተር from 5 prime to 3 prime ነው ግን እዚህ ጋር ኢንዲኬትድ ያለ ነው ካዛክ ፍራግመንት አልታየም ዲያግራሙ ላይ the lagging strand synthesis has fragments ማለት ነው requirements rather than that the magnesium plus is connecting as a cofactor as well as a stabilizing factor the parental DNA template is one of the requirements an RNA primer by the way this RNA primer is synthesized by the enzyme called primase ይባላል RNA primer ለምንድን ነው የሚያስፈልገው ቢባል it can be used to provide the 3 prime OH group for starting the synthesis of the new DNA strand based on that one ከ3 prime OH ላይ ተነስቶ እየሰራ ሄዳል ኋላ DNA chain synthesis ካለቀ በኋላ እቺ RNA primer ትነሳለች ከDNA strand ላይ there are also other proteins that I told you leading strand from 5 prime to 3 prime continuous strand ነው straight ነው the lagging strand has a Okazaki fragments and there is also a difference on the level of replication or origin of replication as well. Misali most of unicellular organisms the bacteria or virus eukaryotes ላይ ማለት እንችላለን they do have a circular DNA structure and they can be able to form only one replication fork or replication bubble that means they might have one origin of replication however when you come to the eukaryotic cells there are a lot of replication bubbles and replication centers ማለት ነው the replication center is a place where the replication starts ማለት ነው unwinding የሚጀምርበት ነው so this is the difference when you see the formation of replication fork when the two strands unwind and separate they form a V shape no? and in one replication bubble there are two replication forks no? ሁለት ሹካዎችን አስቡትና ኦፖዚት ዳይሬክሽን ዛችሁ እንዲስገጥሙት ሙሉ አይሆንም በብል ይሰራል መሃል ላይ ይለያያል so this region is called the replication fork and it moves along the DNA molecule as the synthesis occurs and we can call it the DNA replication process is a bidirectional as we said before the leading strand towards the center of the fork the lagging strand synthesis away from the center of the fork it is two-dimensional and there are requirements of proteins for undergoing the replication bubble the initiation is taking place by the DNA A protein the DNA A protein can be used to indicate the AT-rich regions for a formation of the bubble then in that region it can be used to identify the center of replication center one ለመለየት ያገለግለናል ይለያታል if once the center of replication is identified there are other proteins that are binding with it the initiation of DNA replication requires the recognition of the origin of replication and there are by a group of proteins that form repriming complex these proteins are responsible for maintaining separation of the parental strands unwinding of the double helix as well as for protection of the supercoiling of the double helix of DNA because the supercoiling has its own relationship with the coiling relations ስላለው unwinding ራሱን እንቅፋት ስለሚሆን so DNA protein as I told you before it can be used to indicate the origin of the replication AT-rich regions and DNA A protein is ATP dependent because after it identifies it can undergo this unwinding of the DNA strand in that region after unwinding is taking place there are additional proteins called what we call them the single strand binding proteins or SSB. It becomes binded with a single strand in order to protect the winding of the DNA strand. ራሱ single strand እንዳለ ሆኖ ለመጠበቅ until the replication process is taking place ማለት ነው. Then the other major enzyme after identification of the center of DNA replication is the DNA helicase. Helicase ማለት chain ከፋች ማለት ነው. ሃይድሮጅን ቦንዶቹ ማለት እየቆራረጠ ነው የሚሄደው. Can be used to unbind the double DNA strand and it will form single strand DNA. እዚህ ጋር በቢጫ የምትመለከቷቸው በእንቁላል ቅርጽ ያላቸው single strand ጋር bind የሆኑት those are the single strand DNA binding proteins and they help us to keep the two strands of the DNA separate. And the other one, the DNA helicase has movement. Okay, movement አለው እየከፈተ በሚሄድበት ጊዜ ለዚህ movement ደግሞ ምን ይፈልጋል? It requires energy from the breakdown of ATP. Hydrolysis of ATP ይጠቀማል ማለት ነው. As you can see here, ATP is utilized by the DNA helicase and ADP and PI are formed. And when the third phosphate is released from ATP, it provides high amount of energy for a movement. The other one, DNA B protein is the principal helicase for replication in E. coli. Escherichia coli ላይ የዲኤንኤ ሄሊኬዝ አክቲቪቲ ያለው ዲኤንኤ ቢ ፕሮቲን ነው. So those are the major proteins which are responsible for at least unwinding of the DNA strand. There are other proteins and enzymes. Single strand DNA binding protein, I told you it binds with a single strand, and the binding of the single strand binding protein is generated or activated after the helicase is unwinding the chain. They bind cooperatively, and those proteins are not enzymes, and their major function is to keep the single strand DNA protein from the winding, until the DNA replication process is taking place. Rather than that, the single strand binding proteins have a capacity to protect the single strand of DNA from the attacks by the nucleases. So after the replication process, this single strand binding protein will help for the reassociation of the proteins. Rather than that, during this process, the supercoiling should be also solved. Supercoiling, because supercoiling has its own effect for the coiling. ለምሳሌ አንድ ገመድ ወይ ክሊዛችሁ ዝም ብላችሁ ተጠቅልሉት የተቀላላችሁ ስትሄዱ መጨረሻ በጣም ሲበዛ መጠቅለል ምን ይሆናል? ደብል መጠቅለል ይጀምራል። ዛት የስልክ ገመድ ላይ ዋይሮች እና ያኛው ደብል መጠቅለሉ በመፈቱት ጊዜ it has its own effect for separation of the coiled structure. ስለዚህ prevention of supercoiling has its own effect for prevention of coiling. ስለዚህ supercoiling is avoided during the DNA replication process by a protein called or an enzyme called topoisomerase. Their function is for protection of supercoiling. Now, when we see the enzymes and protein factors which are participating in the process, first one, DNA protein. Its function is, as I told you, to recognize the origin of replication. DNA B protein, the E. coli, it has an activity of helicase ብለናል. coiling prevent ያደርግልናል. opens double strand DNA. DNA C protein, it assists DNA B binding. Okay, the DNA polymerase is a major enzyme which undergoes the elongation process. ገንቢ የምንለው ነው። It has different types, DNA polymerase 1, 2, and 3. We will see them with their specific activities. The DNA G protein, or primase ቅድም ያልኳቸው ካሬቶች ላይ ብዙ ጊዜ primase ነው ያለው. It can be used to synthesize an RNA primer on the DNA strand. Single strand binding proteins, they can be used to keep the two strands as separately as a single ብለናል. DNA topoisomerase prevents supercoiling. So when we see the mechanism of DNA replication, it is catalyzed by DNA polymerase, which needs an RNA primer. ለማስጀመር free 3' OH group ይፈልጋል. So that 3' OH group is obtained from the RNA. As I told you before, and as you can see on the screen by the pink color, first RNA primer is synthesized in a complementary manner with the parental DNA by the primase enzyme. Then the DNA polymerase can get the 3' OH free end to undergo the continuous DNA synthesis from 5' to 3'. So when we see the mechanism, the nucleotides are added by complementary base pairing with the template strand. Substrates, as I told you, deoxyribonucleotide triphosphates are there. They are hydrolyzed to deoxyribonucleotide monophosphate and pyrophosphates. And based on breakdown of the pyrophosphates, they will get energy to form a bond, the histone linkage with the growing chain of the DNA ማለት ነው. Generate ያደርጋል. ፓይሮፎስፌቶች ይወጣሉ. growing chain in a complementary manner with the parental DNA ያደገ ይሄዳል. But always the synthesis is taking place from 5' to 3' direction. እዚህ ጋር እየገነባ ሲሄድ DNA polymerase ባይዘው የተንሸራተተ ነው የሚሄደው parental chain ላይ እየገነባ ሲሄድ it has a proofreading activity from 3' to 5' direction, while it undergoes synthesizing from 5' to 3' direction. So, as I told you before, the DNA synthesis is carried out, and this daughter strand is now completed. You can see on the screen. DNA polymerase 1, it can degrade an RNA primers and replaces it with a DNA nosica. No, DNA polymerase has 3 major types: type 1, type 2, type 3. One of the functions of DNA polymerase 1, it has a 5' to 3' exonuclease activity. Endonuclease activity ማለት 5' to 3'. So እዚህ ጋር እንደምታዩት after the daughter strand is completed the synthesis, the DNA polymerase 1 can undergo the removal of the RNA primer from the newly synthesized strand. Then rejoining of the remaining strand can be taking place by the ligase enzyme. So when we see the replication overall direction, on the fork number, then here there is a helicase enzyme. It undergoes the opening of the chain. Leading strand towards the direction of the replication fork center. This is leading strand from 5' to 3'. The lagging strand is away from the replication fork center. You can see here, 5' to 3' direction. And the lagging strand, as we said before, is synthesized in a fragment form. So in the lagging strand, RNA primer gives 3' end, and after some fragments, another RNA primer is required to provide another 3' end. So the fragments are formed, so Kazakh fragments. But at the end, as we discussed before, the RNA fragments are removed from the newly synthesized strand by the RNA polymerase 1, and the remaining DNA fragments are joining together by the catalytic activity of ligase enzyme. So you can see how the chain is grown. You guys touched number. Chain we are digging here, the parental strand. The new DNA strand is synthesized. So leading strand always synthesis continues in the 5' to 3' direction, and it is continuous synthesis. The lagging strand is also synthesis taking place from 5' to 3'. However, it's a discontinuous with the fragments. So this diagram also is indicating the primer as well as the newly synthesizing DNA strand. DNA polymerase እየተንሸራተተ ነው የሚለው ባይዘው ይሄ ፊልም ላይ ምናምን ወይ ካቦ ተዘርግቶ ከአንደኛው ጫፍ ወደ አንደኛው ጫፍ ማለት ላይ ውሃዎች ክፍተቶች ሲኖሩ የተንሸራተቱ የሚሄዱት የለም። DNA ፖሊመሬዝ ልክ እንደዛ ነው። ፓረንታል DNA ይዞ እያመረተ እየተንሸራተተ የሚሄደው ሞዴሎች አንዳንድ 3 ዳይሜንሽናል ሞዴሎች የሚያሳይ ክሊፕ ላይክ ስትራክቸር። ሶ ዚ ዲያግራም ላይ ፕራይሜስ ተታይቷል። RNA primer is there. DNA polymerase 3 is there. It is a major polymerizing enzyme or synthesizing enzyme. ግንበኛ ነው የሚባለው ማለት ነው። ከዛ ፍራግመንቶችን ተጠቅሰዋል። RNA primer as well as leading gate strand as well as the lagging strand synthesis are also indicated for the replication process. So when we see the final steps in the synthesis of the lagging strand segments, you can see here that 3' to 5' direction. This is the normal strand. Okay. And synthesis is taking place from 5' to 3' direction. Now, there are ribonucleotide monophosphates that are released, by the way. DNA polymerase 1 is there. As I told you before, DNA polymerase 1 has an exonuclease as well as endonuclease activity from 5' to 3'. ያ ማለት ከ5' to 3' ዳይሬክሽን ላይ ባለው ቼን ላይ RNA primer ቆርጦ ማውጣት ይችላል ማለት ነው። So as you can see here, the RNA primer that is indicated by the green color comes cut out by this DNA polymerase 1 activity from 5' to 3' direction when the fragment ላይ ማለት ነው. After that, the DNA ligase has a joining up activity by using the energy from a breakdown of ATP. However, DNA ligase, it requires NAD+ as well. NAD+ as a coenzyme. Then the joining up process can be taking place. As I told you, RNA primers in the lagging strand are removed by the 5' to 3' exonuclease activity of the DNA polymerase 1, and those primers are replaced by the new by the DNA, and the RNA polymerase 1 can undergo that one because it has a synthesis capacity from 5' to 3' direction as well. And the remaining, the gap is joined up together, or the nucleotides are joined up together by the DNA ligase activity we have seen. By the way, we should concern on that. We said DNA polymerase 1 has 5' to 3' exonuclease activity as well as synthesis activity. But it may not have exonuclease activity from 5' to 3' direction of the parental, from 3' to 5' direction. It may not have that activity. From 5' to 3' ካለ ሊያደርግ ይችላል. From 3' to 5' ግን የለውም. So as well, when you have seen the replication, the steps are DNA unwinds and unzips. Step 2, once the molecule is separated, it copies itself, and there are a lot of proteins participating in this process. What I told you here, the DNA polymerases, they vary greatly in processivity, we said. Some add just a few nucleotides before dissociating. The others add many nucleotides, and they do have different capacity based on the rate, based on their efficiency also. ከዛ ውጪ ስህተት የሚሰሩበት ሬት ራሱ ይለያያል ከታይፓቸው። ለምሳሌ አሁን ዲኤንኤ ፖሊመሬዝ 1 ብናየው structural gene for this is because all enzymes are proteins except RNA ነው አይደል? ፕሮቲን ስለሆነ it has a gene poly gene ነው። እንደገና ዲኤንኤ ፖሊመሬዝ 2 ደግሞ ፖሊ ቢ gene ነው ሲንተሳይዝ የሚያደርገው። ዲኤንኤ ፖሊመሬዝ 3 ፖሊ ሲ ነው ወይም ዲኤንኤ gene ነው ሲንተሳይዝ እንዲሆን የሚያደርገው። አሁን መልከቱ 3 prime to 5 prime exonuclease activity as well as proofreading activity ስናይ 3 prime to 5 prime exonuclease ማለት በዛ በኩል ቆርጦ ማውጣቱ ማረም ይችላል፣ proofread ማድረግ ይችላል። ሶስቱም አላቸው ዲኤንኤ ፖሊመሬዝ 1 ዲኤንኤ ፖሊመሬዝ 2 ዲኤንኤ ፖሊመሬዝ 3 they do have this activity. When we see the 5 prime to 3 prime exonuclease activity 5 prime ወደ 3 prime ያለውን ስትራንድ ቆርጦ አውጥቶ ሊጠገን የሚያደርግበት አክቲቪቲ ማለት ነው ራሱ ባይጠገን። This activity is provided only for DNA polymerase 1. ለዛ ነው ቅድም እዚህኛው ጋር እንዳያችሁት 5 prime 3 prime exonuclease activity ተጠቅሞ ነው RNA ፖሊመርን ያስወጣው። But the 5 prime to 3 prime exonuclease activity cannot be performed by DNA polymerase 2 and DNA polymerase 3. When we see the polymerization rate, DNA polymerase 1 can polymerize 60 up to 20 nucleotides per second. DNA polymerase 2 can polymerize 40 nucleotides per second. However, DNA polymerase 3 can polymerize 250 up to 1000 nucleotides per second. That's why we are saying DNA polymerase 3 is the major polymerizing enzyme type. So, when we see the DNA polymerase families, E. coli ላይ ለምሳሌ ወደ 5 ፋሚሊዎች አሉት። DNA polymerase 1 አለ። E. coli ላይ function repairs the replication as we have seen before, exonuclease activity ያለው። DNA polymerase 2 functions in DNA repair mechanism. DNA polymerase 3 is a principal DNA replication enzyme as we have seen before. ፍጥነቱም ኳሊቲው ሲንተሳይዝ ለማድረግ ብዙ ኒውክሊዮታይዶችን per second አቀድም ላይ ነው ከ250 እስከ 1000 ኒውክሊዮታይድ per second ሲንተሳይዝ ያደርጋል ፖሊመራይዝ ያደርጋል that means it's very fastest in comparison with the others. DNA polymerase 4 is a function in DNA repairing ጥገና ላይ ይሳተፋል. DNA polymerase 5 functions in DNA repairing such like that of DNA polymerase 4. ነገር ግን ሪፔሪንጋቸው ሊለያይ ይችላል። አንዱ 5 prime 3 prime repairing ሊሆን ይችላል፣ የሌላኛው 3 prime to 5 prime repairing ሊሆን ይችላል። So when we see that replication is a very accurate process. For example, the process with an extraordinary degree of fidelity. In E. coli, when we see that, even if that much speed is there for the DNA polymerase reactivity, a mistake is made only once for every 10^9 up to 10^10 nucleotides added. That means it's too much. ይሄን ያህል የሚሆን ኒውክሊዮታይድ ከተጨመሩ በኋላ ነው ምናልባት ይፈጠር የሚችለው mistake. For E. coli chromosome of 4.6 times 10 to 6 base pairs, this means that an error occurs only once per 1000 or per 10,000 replications. It's very less ማለት ነው. That's why the same thing it's in our body also in animals, but it's much more perfect process. That's why much more of the population has healthy physical status and genetical status also. So during polymerization, not just on the hydrogen bonds that specify the correct pairing between the complementary bases. ማለት አንደኛው ስትራንድ ላይ እና አንደኛው ስትራንድ ላይ ያለው complementary bases ትክክል ናቸው ወይ የሚለውም ይጠናል። The error might involve that one. For example, adenine should be paired with thymine. In one of the sequences on the DNA strand. Adenine, after the replication process, adenine might be paired with cytosine. በዚህ ጊዜ ምን ይሆናል? ትክክል ያልገባውን ኒውክሊዮታይድ አውጥተው because the exonuclease activity is here, especially by DNA polymerase 1, it can release that one and it can be replaced by the complementary base ማለት ነው። ይሄም ራሱ during polymerization it's one of the process ማለት ነው። For example, as I told you before, you can see this DNA synthesis. Parental DNA is there. DNA polymerase 3 is polymerizing. While the polymerization, the error might be made, even if it's very rare. So adenine should be paired with thymine. However, the newly found nucleotide is the cytosine. Then cytosine is a rare tautomeric form of cytidine is a rare tautomeric form of cytosine. No? That pairs with adenine because the condition is rare, as I told you. Then after, the bonding is formed as it is the wrong attachment between adenine and thymine. Before the polymerase moves on the cytosine to undergo another binding process, it has, especially DNA polymerase 1, has, as you say, it has an exonuclease activity. So, the misleaded C prime ends of the growing chain will not be continued because the DNA polymerase 3 blocks the further elongation of DNA and it undergoes the 3 prime 5 prime exonuclease activity to replace the complementary base sequence with another nucleotide. So the DNA polymerase can sliding back and the enzyme can correct the mistake that occurs in the 3 prime to 5 prime direction by using its exonuclease activity. Then it can resume its polymerase activity in 5 prime to 3 prime direction after this mistake has been resolved. So this is the one that I told you. Misleaded nucleotide ነበረ እሱ እንዲወጣ ያደርጋል. Then after it can be replaced by the complementary. ተሳስቶ ነበር ሳይቲዲን ይገባው ከዛ በኋላ ሳይቲዲን እንዲወጣ አደረገ. Then it can be replaced by thymine by the complementary base with the parental strand ማለት ነው. that ተመልሶ the polymerization will be continued ማለት ነው. So proofreading is one of the DNA polymerase activity. That means ማንበብ ማለት ነው. ሰላም ፎቶኮፒ ስናደርግኛ ወደ ቀኝ እየወሰድነው ማሽኑ በግራ በኩል የሚያወጣ ከሆነ ወደ ቀኝ ያነበበ ወደ ግራ ያወጣል ይባላል. Such like that the DNA polymerase 3 is undergoing proofreading from 3 prime to 5 prime direction. That means the proofreading of the parental strand. And it undergoes synthesis from 5 prime to 3 prime direction. So we can call it the proofreading is not simply the reverse of polymerization reaction because the pyrophosphate is not involved. Then DNA polymerase 3 is holoenzyme. It's replicase complex. They say that it has a lot of subunits. እንዳልኳችሁ ይሄ በገመድ ላይ ክሊፖች ነው እየተንሸራተቱ ካንዱ አካባቢ ለአንዱ አካባቢ ሲሄዱ ታስ ክሊፕስ አይደል such like እንደ ክሊፕ የሚያገለግሉ ሰብዩኒቶች አሉ. ለምሳሌ ቤታ ሰብዩኒት አለ ኤፕሲሎን ሰብዩኒት as you can see on the screen, theta subunit, the delta subunits, the pi subunits, the psi subunits, like subunits. The beta subunit has a major slide clamping capacity. ለማንሸራተት የተንሸራተተ እንዲሄድ ነው parental DNA strand ላይ የሚያገለግለው. ሌሎችም የየራሳቸው ፋንክሽኖች አላቸው እዚህ ጋር የምናየው. For example, alpha subunit can be used for 3 prime to 5 prime profile reading and exonuclease activity, no? And the alpha and epsilon subunits, theta and pi subunits, they are the core subunits, up to the theta and pi. So alpha subunit is a polymerization activity. Epsilon subunit has 3 prime to 5 prime proofreading and exonuclease activity. the theta subunit doesn't have a known function. The pi ላይ ስንመጣ stable template binding ነው እና coenzyme dimerization ላይም ያገለግላል። ጋማ subunit for example clamp loader ነው። clamp ማለት መንሸራተቻዋን እንድትጫን ከስትራንዱ ጋር ባይንድ እንድታረግ የሚያገለግል ነው። ሲግማ subunit it's clamp opener ነው። መያዣዋን ለመክፈት ነው የሚያገለግለው ከዲኤንኤ ባይንዲንግ ጋር እንድትያዝ። ምክንያቱም ቅድም በክላምፕ ስትራክቸር ነው እዚህ ዲያግራሙ ላይ ያየን። then the sigma prime subunit is acting as a clamp loader, the binding loader. The X subunit it can be acting as interaction with a single strand binding protein because the DNA strand is after the opening of the chain, the strand is bound to a single strand binding protein for maintaining the strand as a single. The psi subunit, the interaction with gamma and X subunits, the interaction, beta subunit, the DNA clamp required for optimal processivity. Processivity. So those are their own function. When we see the chain elongation, prokaryotic and eukaryotic DNA polymerases elongate a new DNA strand, said by adding deoxyribonucleotides, one at a time, and to the 3' end of the growing chain, since the process is taking place from 5' to 3'. The sequence of nucleotides that are added in the dictated by sequence of the template strand and which the incoming nucleotides are paired. The instruction or the sequence nucleotide arrangement is obtained from the parental DNA strand. Since the DNA polymerase undergone the 3' to 5' reading activity. ዛና ፕሮፍሪዲንግ ያስፈልገው ያነበበ ኮምፕሊመንታሪውን ለማዘጋጀት ነው። ፓረንታሉ ያነባል ኒውሊ ሲንተሳይዝ ላይ ኮምፕሊመንታሪውን ያዘጋጃል። DNA polymerase 3 as I told you before, DNA chain elongation is catalyzed by DNA polymerase 3. It uses the 3' hydroxyl group of the RNA primer because RNA primer is the one which can be used to start new DNA synthesis on the DNA structure. And the acceptor of the first deoxyribonucleotide is an RNA primer. So DNA polymerase 3 begins to add nucleotides in such a direction to increase the chain of the newly desynthesized DNA. DNA polymerase 3 is a highly processive enzyme, and that is, it remains bound to the template strand as it moves along. It does not have to diffuse away and rebind before adding each nucleotides. So the processivity of DNA polymerase 3 is the result of one its beta subunit forming a ring that encircles and moves along the template strand of the DNA. In this serving as a sliding DNA clamp ነው ቅድም እንዳየነው የተንሸራተተ መሄጃ ነው የሚያገለግለው ወይ ፊልሞች ላይ የተለያዩ ቦታዎች ላይ ኮንስትራክሽኖች ሲኖሩ ከአንደኛው ከፍታ ቦታ ወደ አንደኛው ከፍታ ቦታ ለመሸጋገር ካቦችን ዘርግተው በክላምፕ የተንሸራተቱ ይላሉ አይደል? It might be based on gradient or it might be by በፎርስ ሊሆን ይችላል ኤሌክትሪካል ፎርስ ሊሆን ይችላል ይሄ በክራውን የሚያሽከረክረው ወይ ግሬዲየንት ሊሆን ይችላል ከከፍተኛ ወደ ዝቅተኛ ቦታ ከሆነ ራሱ እየተሽከረከረ ይሄዳል ሶ ያ ማለት እንደ ክላምፕ አለችው ማለት ነው ይዛምት ሶ DNA beta subunit polymerase beta subunit character as a clamping unit ነው to move along the parental DNA strand in order to undergo the proofreading activity. The new strand grows in 5' to 3' direction and anti-parallel to the parental strand that is present in 3' to 5' direction. And the nucleotide substrates are 5' deoxyribonucleoside triphosphates. Okay. So those undergo the pyrophosphate group removal or pyrophosphate bond breakdown to generate energy. And the monophosphate will be added on the growing chain by the energy that's obtained from the breakdown of the pyrophosphate group. So when you can see on the screen now, DNA topoisomerase 2. Sorry, can we make a break or can you continue with this?
23 ስላይድ ነው አይደል ይቀረን ሸር ችግር የለውም እሺ ረፍት እንውሰድ አንድ 10 ደቂቃ
ኦኬ በዚህ ምን ጨርስ ካላችሁ መጨረስ እንችላለን which one is better for you
እንጨርስ ይሻላል እንጨርስ
ኦኬ
ፈተና ስላለ ለሱ ማንበብ እንድንችል ብንጨርስ ይሻላል አሁን
እሺ So the DNA topoisomerase 2, or it has another name, the DNA chiasm. It can be used to protect the supercoiling ብለናል እዚህ ጋር ያለው supercoiling ለመከላከል ነው DNA B or helicase it is a major unwinding enzyme of the double strand of DNA ብለናል indicated ሆኗል DNA primase is there. The DNA primase can also be used to synthesize RNA primer on the growing chain like ብለናል RNA primer one single strand binding proteins አሉ they can keep the strand as a single ሌላው leading strand synthesis እንደሚያነው ነው toward the center of the replication fork it's done by DNA polymerase 3 ነው major polymerizing enzyme ነው ብለናል lagging strand line synthesis are away from the center of replication fork. So lagging strand, there are fragments. Fragments. A diagrammatic expression can increase your memorization capacity. Endonucleases versus exonuclease activities. DNA polymerase 1 has an exonuclease activity. ያለን ስንነጋገር ነበር endonuclease እንላለን endonuclease or exonuclease migrating from 5 prime to 3 prime or migrating from 3 prime to 5 prime so what exonucleases but exonucleases cut from the end of the chain releasing single nucleotide for example it might be from the 3 prime side ሊሆን ይችላል it might be from 5 prime side ሊሆን ይችላል however it should be at the side however if it is endonuclease activity that enzyme has potential to release out the unwanted nucleotide sequence from the center of the DNA strand and to replace with that one inside part, internal part an endonuclease and exonuclease activity. DNA ligase, I told you before also, the final phosphodiester linkage between the 5 prime phosphate group on the DNA chain synthesized by DNA polymerase. We said that. In the 3 prime hydroxyl made by DNA polymerase 1 group on the chain is catalyzed by DNA ligase. ምክንያቱም RNA primer 3 prime in the free 3 prime in the ይሰጣል ከዛ በኋላ especially the lagging strand synthesis fragmenቶች አሉ ብለናል እነዚህ ፍራግመንቶች ድጋሚ ኋላ ላይ ያያዛሉ by the activity of ligase ብለናል አይደል so the RNA primers will be released as we discussed by the DNA polymerase 1 exonuclease activity from 3 prime to 5 prime አዲስ የሚሰራው እዛ ላይ ስለሆነ ከዛ በኋላ the remaining phosphate in 3 prime OH end should be joined together by the phosphodiester linkage. This rejoining is catalyzed by the DNA ligase enzyme. And this process requires energy and removal of the water molecule. Energy or bond formation with the energy ስለሚፈልግ ATP breakdown AMP plus PPI as I told you before. Then the energy obtained from this reaction can be used for the joining of the link. So the joining of the two strands of DNA requires energy. So there are different models of DNA replication, which is also one of the DNA replication model. It can show different proteins participating in this process. This is what we discussed already, but I included a lot of diagrams. Whenever you will see different types of models, it will increase your memorization capacity and understanding of the replication process. We said that some microorganisms, like the E. coli, they do have a circular DNA structure, we said. So they do have various types of proteins also to undergo different process. For example, they have the TOS protein that arrests replication for motion. That means they make a blockage for the replication process that's taking place. And they are occurring this on the opposite side from the origin of the replication. And they are flanked by 6 nearly identical non-palindromic or 23 base pair terminator T sites. ማለት ምንድነው? they are acting እዚህ ጋር እንደምታዩት የተለያዩ ጂኖች አሉ። ፕሮቲንስ አሉ ሶሪ ያውን እነዚህ ለምሳሌ ዘቱስ ሞኖመርስ ናቸው። ያንዳንዱ ግን የተለያየ ታይፕ አላቸው። ቲኤፍ ቲአርቢ የሚባሉ ኮላይ ክሮሞዞም ላይ ያሉ የተለያዩ የጂን ፕሮቲኖች ናቸው። ሶ ይሄኛው አሁን ቱስ ፕሮቲንስ የሚባሉት ለምንድን ነው በዋናነት ያገለግላሉ ነው ያልናቸው? they are acting as a signal for stopping of the replication process. በቂ የሆነ ዲኤንኤ እንደተመረተ ያ replication process አብቅቶ parental DNA ሼፑን እንዲዝና ደብል ስትራንድ structure form እንዲያደርግ ማቆም replication termination of replication signal ሆኖ ያገለግሉናል ማለት ነው። eukaryotic DNA replication it's mostly a similarity except some differences with the prokaryotes. Similar to that of the prokaryotes, and differences in the process are related mainly to the vastly larger amount of DNA in the eukaryotic cells. You know, I told you that even the center of replication or the forks or the bubbles of replication are much more in the eukaryotes as the eukaryotic cells, including us. We have a lot of genes having long DNA structures. Association of eukaryotic DNA with histones is nucleosome. The way prokaryotic cells, they do not have a specified membrane for each of subcellular organelles. We do have a specified membrane for each of subcellular organelles, including the nucleus. And this DNA replication process is taking place in nucleus. That's why the histone proteins are also present in the eukaryotic cells. However, histones are not present in the prokaryotic cells. Eukaryotic cells have multiple origins of replication. The single origin of replication in prokaryotes. Eukaryotic single-strand DNA binding proteins and ATP-dependent DNA helicases are analogous to those of the prokaryotic enzymes, and they do have similarity. And the RNA primers, they are removed by RNase H in the prokaryotes. And FAN1 types of protein stabilities are there rather than DNA polymerases. Those are also proteins and enzymatic activity. The eukaryotic cell cycle, it's known that the events surrounding eukaryotic DNA replication and the cell division are there, and the mitosis process is there, and they are coordinated to produce the cell cycle. So, as you know that in the eukaryotic cells, the cell cycle has different phases. In the first phase, 3 phases as orders are G1 phase is there, S phase or synthesis phase, and the G2 phase, and they constitute interphase, And the cells spend most of their time in the 3 phases in order to undergo the mitosis process and carrying out their normal metabolic activities. In the 4th phase of is the mitosis, and the process of cell division can be taking place on the 4th stage, as you know. In which stage of the cell cycle is the DNA replication occurs? It occurs in the synthesis or S phase. As you can see on the diagram here, the G1 phase, growth and metabolism can take place. On the S phase, DNA replication can take place. The other G2 phase can undergo preparation of the cell division. In the last mitosis phase or 4th phase, in the mitosis and the division into 2 types of cells can be taking place. So S phase is responsible for undergoing the DNA replication. Points of origin of replication. In contrast to bacterial chromosomes, eukaryotic chromosomes have multiple points of origin at which the replication begins. We have seen it in the diagram. They do have also multiple bubbles appear as points as a chromosomes. And at each end of the bubble, the replication fork forms. We already discussed that. And thus each bubble has 2 forks. The 2 forks are combined together in order to form a bubble, the left side fork and the right side fork. And the DNA synthesis occurs at each fork. So when we see DNA polymerase of eukaryotes, DNA polymerase alpha can initiate the replication process. And it can synthesize primers in DNA G and primase activity have the effects complementary to the DNA polymerase alpha. And see DNA polymerase beta unit can undergo replication with low fidelity and repairing capacity it has. DNA polymerase gamma that we have seen before, it can undergo polymerization in the mitochondria, since the mitochondrial DNA has a circular structure, we said. DNA polymerase sigma, it can undergo elongation, or we can call it has similar property, or another name, we can call it DNA polymerase 3. DNA polymerase epsilon has a proofreading and gap filling activity. That means it is DNA polymerase 1 below. It has higher activity of DNA polymerase epsilon subunit. So initiation, the eukaryotic origins are shortened than the prokaryotic, or we can call it from the E. coli, as it is a major example for the prokaryotic organoids. And the requires DNA polymerase alpha has primase activity, can be used to synthesize RNA primer on the growing strand. DNA polymerase sigma has polymerase activity and helicase activity, we said that. So it can undergo unwinding the double strand DNA as well as undergoing the DNA polymerase reactivity or polymerizing activity. So they need topoisomerase and replication factors to assist. So there are different replication proteins that we have seen. We can call them the replication factors. When you see the elongation process of the DNA replication in nucleosome assembly occur simultaneously. In the overall replication speed is compatible with that of the prokaryotes. just leading strand line, lagging strand line, ሁሉም ፕሮቲኖች ፓርቲሲፔት ይሆናሉ። But majorly in this diagram, it's showing the single strand binding protein. It can be used to protect the attack by hydrolysis or nucleases plana. So it can be used to keep the strand as a single. So eukaryotic DNA polymerase, they do have different subunits. Functionation, ቅድም በቴብል ላይ አይተነዋል። subunitoቹን at polymerase sigma, major replicative enzyme. polymerase alpha and polymerase epsilon are involved in the replication, even if they are not major replicating subunits. Polymerase beta and epsilon, as well as polymerase alpha, they appear to be involved in the DNA repair process. That means DNA polymerase 1 has much more active those subunit activities. Polymerase gamma is located in the mitochondria and replicates the DNA of its organ in a circular DNA. So the polymerase subunits are there which lack the 3' to 5' exonuclease activity. They used when DNA is damaged in the remaining subunits. So the table literally shows the activity of functions of eukaryotic DNA polymerase. Polymerase alpha, it's replication, and the replication in a complex with primase and S in starting the print, but it doesn't have exonuclease activity. It has a DNA repairing capacity, like the beta polymerase beta subunits. It has DNA repair exclusively, but it doesn't have exonuclease activity. Polymerase gamma subunit has DNA replication in the mitochondria, as we said, the functional. Exonuclease activity 3' to 5' direction, the parental DNA. Right? Polymerase sigma replication, no? Processive DNA synthesis on leading and lagging strand. That means, as we said before, DNA polymerase 3 has better sigma subunits. ምክንያቱም ብዙ ፕሮፐርቲያቸውን ያየነው ይሄ ዲኤንኤ ፖሊመሬዝ 1 2 3 በስካርቻኮ ላይ ስለነበር ብዙ መመሳሰል ስላለው ነው ኳሊቲክ እና ፕሮካሪዮቲክ ዲኤንኤ ሲንተሲስ ልዩነቶች እንዳሉት ሁሉ ሶ the polymerase sigma subunit has 3' to 5' exonuclease activity. Polymerase epsilon subunit has also replication capacity, but the replication is taking place in some tissues. And it has also 3' to 5' exonuclease activity. It has also the DNA repairing capacity. Other subunit polymerase kappa and so they all have repairing capacity, however, all of them will not have the exonuclease activity. When you see the eukaryotic replication complex, those all the proteins that we have mentioned are combined together. The subunits have their major own function, we said. And many proteins bind at or near the replication fork and participate in the process of replicating DNA. And the polymerase sigma is the major replicated enzyme, already mentioned on the table. However, before it acts, the primase activity should be required. And this primase activity is associated with the polymerase alpha subunit. It can be used to synthesize the RNA primer to provide 3' OH group for the growing DNA strand. And polymerase alpha is a major polymerizing enzyme with DNA polymerase 3 activity. It adds about 20 deoxyribonucleotides to this RNA. And it can dissociate from the template. In most of the conditions, the RNA primer that do have 20 ribonucleotide sequences is enough to initiate the synthesis of the new DNA, or enough to initiate the DNA polymerase activity. Okay, and the polymerase alpha has a low processivity, no? Primase activity can be synthesized RNA primer. On the leading strand, polymerase sigma deoxyribonucleotides to RNA DNA primer. It can undergo the major synthesis process or DNA polymerase activity. So polymerase sigma is highly processive enzyme, and it has similar property with what we mentioned as DNA polymerase 3, major polymerizing enzyme. So this table involves the major proteins involved in the replication process as revision ማለት ነው። እስካሁን ላየናቸው revision. So the DNA polymerases, they are nucleotides to a strand growing from 5' to 3' direction. The DNA polymerases copying a DNA template from 3' to 5' direction. Remember the last one, proofreading from 3' to 5' direction. Based on that proofreading, the complementary bases are synthesized in the 5' to 3' direction. In the primase, it synthesizes RNA primers for providing free 3' OH group for the growing chain of the new DNA synthesis planner. Helicase is one of the unwinding enzyme. Separates the parental DNA strands and unwind the double whole helix. Single strand binding proteins, they prevent the single strand of the DNA from reassociating, as well as from the attack from the nucleus. Topoisomerase relieve functional strain or parental duplex caused by unwinding. That means they prevent the supercoiling of the DNA strand. And the enzymes that remove the primers, RNase H enzyme, with H7 that can hydrolyze RNA of the DNA-RNA hybrid on the leading strand as well as the lagging strand. Flumpindol nuclease 1 or FAN1 that we mentioned in the Scharcher coli blank. It has exonuclease active such like that of DNA polymerase 1 ብለናል። ሌላኛው is the 5' end of primer. It undergoes the cleavage, and it also undergoes the cleavage in the DNA region of the primer. DNA ligase, I told you that it can be used for joining up the separated DNA strands together by using the energy obtained from the ATP breakdown. Other the recent year, the telomere. Telomeres, they are very important terminal structures of eukaryotic DNA chromosomes. And they are composed of terminal DNA sequence in the protein. When you see the function, no chromosome chromosome in the cylinder አይነት structure is ቢኖሩ እዛ ውስጥ ብዙ ዲኤንኤዎች ነው የተጠቀለሉ የሚገቡት። ቅድም የነገርኳችሁ የሜትሮ ኤግዛምፕል የምትጠቀልለው ሜትር ጫፍ አለች አይደል? ጫፏ ባትኖር ምን ትሆናለች? ውስጥ በቃ ይገባል፣ ለማግኘት ይከብደናል አይደል? የሚጠቀለለው ውስጥ ይገባል። Such like that, for the DNA replication process, the DNA stored in the chromosome can be available from the chromosomes when the tips telomeres are available in required length ማለት ነው። ጫፉን ነው የሚያገለግሉት፣ በእነሱ ተስቦ ይወጣል፣ ሪፕሊኬሽን ለማካሄድ ይቻላል። ስለዚህ ውስጥ ዲኤንኤ ክሮሞዞም ውስጥ ያለው አንድ ላይ ተጣብቀው ውስጥ እንዳይቀሩ ለማድረግ ይረዱናል ማለት ነው telomeres. So the terminal structure of eukaryotic DNA of chromosomes, they are composed of terminal DNA sequence and they do have also proteins. So their function majorly is to keep the termini of the chromosomes in the cell from becoming entangled and sticking to each other. So in the repetitive DNA sequences, most of the telomeres do have a sequence of TTAGG No, invertebrates. እዚች ጋር እዚች ዲያግራም ላይ እንደምታዩት ለጫማ ክር ማሰሪያ ጫፍም ትመስላለች። እንደዚህ ከሚሆኑ ልክ ቴሎሜሮች ሲኖሩ ፍሪ ኢንድ ይሰጡታል and they will make the DNA replication process continued. So in 2009 the ones who identified the activity of those telomeres have got a Nobel Prize in recent years. The band ምንድነው? Nowadays they are saying the telomere length is associated with aging. When the age is increasing, the length of the telomeres becomes reduced. ያጠሩ ይመጣሉ ከዛ ዲኤንኤ መሻለ ኢንታንግል ይሆናል ምንሞትበት ጊዜ ላይ ዲኤንኤ ኢንታንግል ሆነ ማለት the DNA cannot be able to undergo the replication. If the replication is not taking place, translation cannot be occurred, then proteins cannot be synthesized. If the proteins cannot be synthesized because as they do have different functions, it will go into the days. So nowadays they are saying keeping the length of telomeres as usual and their activity is used to increase the age as well as it might protect the days. Some of scientists are saying like this. So telomerase is an enzyme that essentially builds the new telomeres and maintains the integrity of the DNA telomere. And the telomere is composed of the telomerase RNA, telomerase association protein is there, and the telomerase reverse transcriptase activity is there. Since the telomere nucleotide sequences are synthesized from the RNA. So it is able to synthesize DNA using RNA as a template. There's a reverse transcriptase activity as well as synthesis of new DNA from parental DNA. ቅድም ኖቤል ፕራይዝ ዊን ሲያደርጉ የጠቀሱት ቴሎመር ሲንተሲስ ሞዴል it's like that of ኢንክወርም ሞዴል ይባላል። ኢንክወርም ካያችሁት ይታጠፋል መሬት ላይ ሲሄድ ይዘረጋል ይሄዳል ማለት እየታጠፈ እየተዘረጋ ነው የሚሄደው as you can see here on the diagram. Such like that the telomere in the tip telomerase enzyme are there. It uses an RNA primer as you can see here. First the telomere tip undergoes synthesis or elongation on the 3' OH group based on a complementary sequence from the parental RNA. It undergoes elongation እንዳያችሁት ይደጋሉ nucleotide sequences based on a complementary link. For example, if the cytosine is there in the parental RNA molecule, guanine will be synthesized on the new growing telomere chain. First elongation. After elongation, they undergo translocation. Then after translocation, they will undergo new growing of the chain. Then there will be another translocation based on such a type. The telomere nucleotide sequence chain can be grown by the catalytic activity of telomerase enzyme. That's why we say telomerase enzyme has the telomere RNA, which can act as a strand, telomerase associated protein, and the telomerase reverse transcriptase activity, which are important for the synthesis of new telomerase. So significance of telomerase is highly active in the embryo since during development of the fetus. The continuous protein synthesis is required for different structures of the fetus. And after birth, it is active in the reproductive and stem cells since they do need much more fast synthesis of proteins than others. Telomerase may play important role in cell aging and cancer biology, especially nowadays they are doing a lot of studies. When you see the telomerase and senescence, that means a programmed cell days, in most somatic tissues, telomerase is expressed at very low levels or not at all. As cells divide, telomeres shorten. And they are expecting that this is one of the cause for the occurrence of cancers. ብዙ ሴል ዲቫይድ እያደረገ ባለበት ቁጥር ቁጥር ይሄ የቴሎመር ሌንግዝ እየቀነሰ ኋላ ላይ እያጠረ መሄዱ can has its own effect ነው ስለዚህ keeping the telomer's length as such might be a target for the cancer treatment it might be hopeful target ሊሆን ይችላል ይላል so that's why telomeres is as a cancer strong evidence to suggest that the absence of senescence in cancer cells for the programmed cell death is linked to the activation of the telomerase. Telomerase is an attractive target for cancer chemotherapy. That's why we are calling it. So, thank you. ቅድም እንዳልኳችሁ ለእድሜ የሚሉ ጥናቶች አንዳንድ የተያዙ ልታዩት ትችላላችሁ but I think this topic aging and telomerase or is there in your seminar presentation then we'll see it. Do you have any question? ጥያቄ ያላችሁ?
እስኪ ጥያቄ ካለ ቻት ቦክስ ላይ ትንሽ እንጠብቅ ይቻላል።
Okay. Don't you have any question? The difference between DNA's replication and duplication. DNA no? DNA replication and duplication. Replication is there a duplication process? I never seen that and it's not there. We can call it it's DNA replication. The replication has those processes and those rules and regulations. And the duplication, I don't know. Duplicate I know. Synthesis ከሆነ በዚህ way ነው replication. Duplication ከሆነ maybe all nucleotide sequences become similar and it cannot involve this process. But replication like complementary sequence ነው ሲሰራ ምሳሌ እዛኛው ላይ ያለው ፓረንታሉን ታይሚንግ ከሆነ ኒውክሊዮታይድ ሲኩዌንሱ እዚህ ጋር አንድን የሚበት ስራ ይመጣል እንጂ ራሱ ኮፒድ አይሆንም ዱፕሊኬሽን ግን ከሆነ ያኛው ስትራንዱ እንዳለ እስከ ኒውክሊዮታይድ ሲኩዌንሶቹ ሲመጣ ዱፕሊኬሽን ነው it doesn't have any difference with that strand ነው ስለዚህ ሪፕሊኬሽን አይሆንም replication ግን based on the Chargaff's rule based on the complementary rules the synthesis of new DNA from the parental strand of the DNA. Have you got my point? Have you got it? What about others? Do you have any other question? Don't you have it? Okay. Thank you very much. We will continue tomorrow. I will attach both of the slides, yesterday's and today's slides. And yesterday, I already forwarded the recorded video. Today also, I will send the recorded video. Have you got it yesterday?
Yes, አግኝቼዋለሁ ግን እንትን አላለኝም ዳውንሎድ ማድረግ አልቻልኩኝም እና እሱን እየሞከርኩ ነው ካልሆነ I don't know drive ላይ play ያደርጋል ለኔ እንትን ያደርጋል ቪዲዮን ነገር ግን ዳውንሎድ ማድረግ አልቻልኩም ግን ለሞክርና ካልሆነ ቴስት ቱ ባለን ቀን መጥቼ ይቀበላለሁ ወይ በፍላሽ
Okay. Have a nice day. See you tomorrow morning.